guess I'm just a play date to you.
Hello everyone, my name is Abdurrahman Alay and today I'm going to speak about PCR and its applications. Polymerase chain reaction or PCR uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind to or anneal to a complementary matches the target DNA sequences. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the prime sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begin to accumulate. After 30 cycles, as many as 1 billion copies of target sequence are produced from a single starting molecule. And now let's move on to the applications of PCR. So the applications of PCR is used in the identification and characterization of infectious agents. It is also used in direct detection of microorganisms in patient specimens, identifications of microorganisms grown in culture, detection of antimicrobial resistance, investigation of strain relatedness of pathogen of interest, genetic fingerprinting, detection of mutation, cloning of genes, and PCR sequencing. And also, for the applications of PCR, I could say the most important 3 uses are molecular identification, sequencing, and genetic engineering. In molecular identification, it contains molecular ecology, DNA fingerprinting, classification of organisms, genotyping, drug discovery, etc. In sequencing, it has bioinformatics, genomic cloning, and human genome project. And in genetic engineering, it has site-directed mutagenesis and gene expression studies. Thank you.